and my area of interest is like sequencing and analysis of genetic materials.
Okay, all right. So how did you get interested in the research part of it? Do you plan on going for your PhD?
Yeah, in future, yes, of course, I'm going to plan for it. So that's why I'm preparing from now itself.
Okay, all right. Thank you so much, Manjushri. So
Yeah,
let me
thank you.
check. Yes. We have a few more minutes before we start the webinar. So we'll go over to the next participant, Aniket. Hello, Aniket. Aniket, can you hear us? I guess he's not available. All right. So then let's move on to the next person, Saranya. Yes, Dr. Guan, it's nice to see you back. Yes, thank you so much, Dr. Guan, for confirming. Perhaps we can move on to the next participant. Saranya, could you please also introduce yourself and let us know what are your research interests, let us know your education background. I guess there seems to be some network error at the end. But if you don't feel shy or anything, please feel free to introduce yourself. Just be relaxed. It's going to be an interactive session throughout, right from the start till the beginning, till the ending. If you have any questions, you can let us know, and we'll be able to guide you on that. So, all right, let's move on to the next person. If you're not able to unmute yourself, you can introduce yourself in the chat box as well, and I can read out, and then so that we can get familiar. So the next person, Suhel, are you with us? Could you please also introduce yourself? What is your education background? What is your research interest? I can see that a lot of the participants are from the bioinformatics background. Do we have any participants from biology background or computer science background? Okay, sure, Dr. Guan. Okay, no problem. Okay, thank you so much, Dr. Guan. So Dr. Guan says he is from a biology background. And what are your research interests? Can you please let us know that as well? Okay, his interest is in oncology. And we have a couple more participants who have joined. So who would like to go next? Okay, cancer NGS. So thank you. And Abhilashni, how are you? Can you also introduce yourself? You can let us know about your education background and research interest in the chat box. All of the participants, feel free to introduce yourself. And if you have any queries, you can unmute yourself and ask, or you can put them in the chat box and we'll be able to guide you. All right, so we are late by 2 minutes. I'll just quickly start with the, okay, Rizmi, can you try connecting your audio? I can see that the audio is not connected. Let me, Nalika, can you please type in the chat and let her know that she has to connect her audio, because that is why she is not able to hear. All right, so without further delay, I'll just quickly start with the session, but please continue to introduce yourself in the chat box, and let's keep it interactive. All right, so hello everyone, a very warm greeting to one and all. My name is Sri Gauri, and I would like to welcome you all for the webinar on OmicsLogic Multi-Omics Research Program. So this program is a comprehensive research program that covers various aspects of omics data analysis and research, and it's designed to accommodate a wide range of participants from high school students to experienced researchers. And the program offers a hands-on approach to bioinformatics, guiding you from basic data analysis to advanced research methodologies, with even opportunities for publication. And now in this webinar, we will be covering several topics such as key features and associated resources of the research program, introduction to our cloud research infrastructure, help you choose the right specialization track that best aligns with your career goals, an interactive live Q&A session, as well as the various scholarship opportunities that are available to the participants. And before we begin with today's webinar, let me also take a brief moment to introduce you to the team behind the program, since I can see that a couple of participants are new to us and are hearing about the program for the very first time. So we are a US-based bioinformatics company who is working with multiple academic and commercial collaborators to develop easy-to-use analytical tools, and our mission is to make bioinformatics more accessible. And now we have had the opportunity to work with coordinators from different parts of the world to offer bioinformatics training programs, reaching 24K plus participants in different countries across different specializations, including oncology, infectious diseases, precision medicine, neuroscience, and data science. And as a part of the program, you will have access to the OmicsLogic resources, which includes training modules, data analysis tools, curated project data sets, and interactive sessions with the mentors to give the student a clear path of the Bloom's taxonomy pyramid. And now this also comes with an access to our coursebooks, which include courses on bioinformatics, precision medicine, multi-omics, including genomics, transcriptomics, metagenomics, and epigenomics, data science that includes bioinformatics using R and Python programming, as well as cheminformatics, and example projects of different fields which you could learn from and replicate for your research work. Now, the OmicsLogic platform also gives options for analyzing a given data. For instance, you can learn bioinformatics without any coding at all. And for those interested in opting the non-code pathway, they will be given directions to access the T-Bioinfo platform for bioinformatic processing and analysis of data. And this platform includes demo pipeline as well as data management and analysis cloud infrastructure to run the pipeline. So now to view the various courses, example projects, and student projects, we'll first start by signing up on the OmicsLogic Learn portal, and I'll walk you through a brief demo on how you can get started. So here is the link for signing up. Okay, so here, for those of you who are new to this program and you're hearing about it for the very first time, then this is the Learn platform where the participants, once they enroll in the program, they'll be able to access the various training resources that they get access to. So to be able to guide you on how you can get started, you need to first sign up on this portal. To be able to do that, if you don't have an account previously on the portal, you can visit the link that I've shared in the chat box and click on the create an account option that you see here. And once you click on this, you will be asked to enter your name, email ID, and password, as well as several few details such as your university and so on. So you can enter those details and click on submit, and your profile will be created. Or if you already have an existing social account such as Google, Facebook, Apple, GitHub, or Twitter, you can simply directly click on those and create an account linked with your existing social media handle. So with that, I'll just pause for a couple of seconds for the participants to sign up so that I can help you walk you through the different components on the portal where the mentor will be guiding you as a part of the research program in just a while. So let me know in the chat once you have signed up. Please put a yes. And if you already have an existing account on the portal and you are good to go, put a done in the chat. And I have opened my chat box on the side. So I'll just pause for 2 to 3 minutes so that you can find. Okay, Sadhanya has already signed up. Rizmi has also signed up. If you're facing any technical issues while you're trying to log in or creating an account, you can let us know in the chat box, and our technical team will be able to help resolve the issue so that you can access. Well done, Abhlashni. She has also signed up. Thank you, Nikta. All right, Kavya Sudha has also signed up. Sakita has also signed up. All right, Lakshmi has also signed up. So I'll just proceed ahead with navigating and showing you how you can get started. So please feel free to continue to let me know in the chat once you have signed up. And if you have any issues, once again, you can let us know. So I'm going to sign in using my existing social media, existing email ID and password. So here, once you sign up, you will be able to see the very first page that you're seeing is the courses page, right? So before we navigate to that, the first thing that you have to do once you sign up is to complete your profile. So to be able to do that, on the top blue bar, you will see welcome back with your email ID. Click on that and it will take you to your account setting, right? And here you will see a profile button, click on that. And here, the first thing that you have to do is complete your profile, right? Make sure that your name is in proper capitalization and there is no spelling error, because as you complete the courses and you get the certificate, this name will be printed on it. And also add a brief bio about yourself, just your educational background and research interest, and a link to your social media profile. And add a profile picture or your university's logo, and your profile should be showing 100% complete, right? And now if you head to the courses tab that you see here, you'll be able to see the various courses that we provide on the portal, which ranges topics from introduction to bioinformatics, genomics, transcriptomics, metagenomics, epigenomics, as well as case studies on various specialization topics, such as oncology, infectious diseases, and so on. So here in the search bar, if you enter a keyword that you are interested to learn more about, let's say you are interested to learn genomics, then you can simply enter that and it will display the relevant courses and example projects. And now another tab that I want to show you is the programs tab. There you will find the research program that is part of today's webinar, right? And along with that, you'll also see several other training programs in different fields that you can explore at your own pace. So that is all that I wanted to talk about in getting started on the OmicsLogic Learn portal and navigating on the portal. And now to introduce you to the training program, the various features, the associated resources, and various specialization tracks, I'd like to pass on the stage to my colleague, Sonalika Ray, who is also mentors of the program. Over to you.
Thank you so much, Srigari, for the overview, and a very warm welcome to all of you. Good morning, good evening, or afternoon, in whichever part of the world you are. I'm Sonalika Ray, and I'm the bioinformatics project manager at Pine Biotech. And today we are going to talk about the structure of our research fellowship program. We have designed this program to cater to learners from all the backgrounds, and we have organized it into 3 distinct levels to ensure a gradual and an in-depth learning journey. So the question that arises is why you should join the OmicsLogic community and the training and the research program now. What is the need? So to answer this question, in the ever-evolving field of omics research, there's continual advancements in technology and software that has improved processing capabilities, but the data sets under study are often vast and they require substantial computing power and memory as well, beyond what most personal computers can handle. And accessing and working with publicly available data can be complex, typically requiring coding skills, which many biologists lack. Likewise, the analysis pipelines and the programs that are involved often demand coding expertise. And in university settings, the students receive valuable theoretical knowledge in research project design, but they lack the knowledge in research project design or a hands-on experience due to these technical limitations. And the faculty members, often specialists in their own research areas, may struggle to cover diverse topics or unfamiliar technologies outside their expertise. For instance, a professor well-versed in oncogene expression may have limited knowledge of metagenomics in seawater. So to address these several issues that are present in terms of what is there in the academia and how you can work on the research process without being an expert with the coding, we'll try to understand today how we can join the program and learn from this program how we can use the automated pipelines to process or automate our research work and get our results to interpret insights. Spend less time on working with the code block for analysis, but more time on the interpretation. So the OmicsLogic research program is designed to provide an exceptional opportunity for researchers and students to delve into the world of bioinformatics and harness its potential for analyzing complex life science data. And through a well-structured blend of online resources and personalized guidance from expert mentors, the students would engage in diverse omics research projects. Now, who can join the program? So OmicsLogic is an all-encompassing platform designed to meet the needs of a wide range of knowledge seekers. It is inclusive and welcomes everyone, including high school students or graduates, researchers, even individuals from non-bioinformatics backgrounds. So whether you are just starting your educational journey, have a degree in hand, or you are pursuing cutting-edge research, OmicsLogic would offer resources, courses, and tools that would cater to your unique level of expertise and interest. And it's a space where individuals with diverse backgrounds and goals can come together to explore and engage with the exciting world of bioinformatics and omics data analysis. The Research Fellowship Program is designed with 3 progressive levels that cater to individuals of all the backgrounds. So there are 3 levels: beginner, intermediate, and advanced. The beginner level spans around 2 months. It provides foundational knowledge with predefined research questions and project reports. The intermediate level would last for 3 months, and it will let you propose your own research question and work on a dissertation. Finally, the advanced level spans around 6 months that equips you with advanced data science tools and techniques to address your research question, aiming for research publication. And this inclusive program would welcome high school students, graduates, researchers, and those without a bioinformatics background, ensuring that anyone can embark on a rewarding research journey. I'll take you through each one of these levels in more depth, and we'll try to understand what are these different levels all about. Before I get on to discussing about these different levels, I wanted to discuss about the different research specialization tracks that you can work on in the Research Fellowship Program. So there are several tracks that you can work on. One of them is oncology. Oncology research focuses on cancer-related studies and therapies. Then you have precision medicine that emphasizes personalized healthcare based on individual genetics and characteristics. Then you have infectious diseases that explore diseases caused by pathogens such as bacteria, viruses, and fungi. Then are metabolic disorders that encompass research on conditions like diabetes and obesity. Then you have neuroscience that investigates the brain and the nervous system to understand the cognition and disorders. And then there are fields like system biology, agriculture, space omics, and chem informatics. System biology studies complex biological systems and their interactions. Agriculture research centers on improving crop yield and sustainability. And space omics examines omics data in space environments for space exploration, a very interesting field that's coming up. And then you have chem informatics that focuses on computational methods for chemical research. We have met a lot of research fellows, and some of them come with an idea what they want to work on, a topic or a field that they want to work on with, and different types of data that they want to explore. But we also meet a lot of newbies who are not sure or not clear which specific path do they want to select. That's why we wanted to discuss about the different tracks that you can pick your research question from and get started. Now there are different types of research specialization tracks that I just discussed and what type of study is possible under the track. But I also wanted to discuss what type of data for all of these different tracks can you explore. So in your research journey, within the various specialization tracks that I just discussed, you can harness the power of diverse omics data types. There are fields called, the generalized word is omics, that is collection of something. You have phenomics, genomics, epigenetics, transcriptomics. There are also fields like single cell transcriptomics, metagenomics. So genomics allows you to delve into the genetic makeup of organisms, uncovering DNA variations that are crucial for precision medicine, agriculture, and space exploration. Then you have transcriptomics that provide insights into the gene expression patterns, shedding light on the molecular mechanisms behind the diseases, cellular diversity, and responses to stimuli. Single-cell transcriptomics will take a more granular approach, examining individual cell behavior that is vital for understanding complex systems, such as the nervous system or tumor microenvironments. And then you have epigenomics that explores modifications to DNA that impacts gene activity, aiding research in areas like metabolic disorders and neuroscience. Lastly, metagenomics that is invaluable for investigating the microbial communities, whether in the human gut, soil ecosystem, or infectious diseases, offering insights into the interplay between hosts and their microbiota. Now, these omics data types provide powerful tools to explore the intricacies of your chosen specialization and uncover novel insights. Now, many of you might be thinking that you don't have a background in omics field. If you are from, I heard the participants telling that they are from biochemistry or biotechnology or microbiology, but not the core bioinformatics. Those who are from the core bioinformatics field, they might have dealt with such type of omics data. But for the wet lab students, you need not worry. We'll take you through a training process. We'll teach you a specific type of omics analysis that you're interested to work on with, and then take you a step ahead with your research. I'll discuss what the process looks like in my next slides. So that was about the research specialization tracks that you can work on. Obviously, one track that you need to pick based on your research interest, but those were the possibilities from where you can pick your specific research interest and the different types of data that you can work with. All these different types of data could be used for each and every specialization track that I previously mentioned in the previous slide. So I mentioned that we have three different levels: the beginner level, the intermediate level, and the advanced level. Now, these levels definitely vary in terms of duration, in terms of the project, the research project question, in terms of the outcomes of the program, but also with the program, you have a specific curriculum that we have put together to teach you while you're working on your research project. Because as you learn, only then you can implement those things or those concepts for your research work. So the first level is the beginner level. I'll take you through very briefly about these different levels. It's very important for you to understand what each level caters so that you can pick the level that works for you at the moment. So the beginner level has 6 specific sessions that you need to cover in the initial phase of your research work, the moment you join the program, and then you start working on your research question. In the first session, we will discuss about how to identify the research gaps and formulate research questions. You'll learn how to conduct a literature review to build a strong foundation for your research. In the second session, that is the research proposal and research methodology, we'll dive into the process of creating a research proposal that outlines your project's objectives, significance, and methodology. Understanding the different research methodologies and how to choose the most suitable one for your study will be the key focus here. And then you have the third session, that is data analysis and the project timeline. So this session will cover the basics of data analysis techniques. We'll also discuss how to create a project timeline, ensuring you stay organized and meet your research milestones. The fourth session would be around budget and resources, ethical considerations, and project management. This session is very important for those who are working on a research work in an institution. It could be PhD scholars or researchers. I won't say this session would be important for those who are in their graduation or post-graduation, but yes, go through the session so that you know what steps to consider in future. So in this session, we'll discuss about budgeting, resource allocation, and the importance of financial planning for your research. Ethical considerations in research are critical, and we'll explore the ethical guidelines as well that you need to follow. You'll also learn about effective project management to keep your research on track. This is one of the very important things that I feel every student, be it a graduate, postgraduate, or a researcher, should have a skill of project management. Because I've seen a lot of students working on their project and missing out on their deadlines and not meeting their milestones on time. And that really messes up with your project because the last hour you just rush and put together everything, not making complete sense of your results. Also, I've seen that because you don't have these project management skills, you work a lot, but you're not able to put together a good, meaningful presentation to showcase the work you really did for months. So this is what we'll discuss next. The next session is on data collection and validation and data interpretation. So this session will focus on collecting and validating data for your research, and we'll discuss methods for data interpretation and how to draw meaningful conclusions from your findings. We'll discuss somewhat about the visualizations. We'll also cover about visualization in detail in the sessions though, but this would be an overview. And finally, project presentation. Project presentation is a skill to put together everything that you worked on for months and months, to put it up together as a story in those slides and present what you worked on, what methodology you opted, what results you obtained, and then how do you conclude your presentation is very important. So we'll discuss how to combine your project that effectively communicates your findings. And we'll also cover the process of submitting your research project on the portal. So that is what the beginner level research fellows will learn. Now, those who join the intermediate and the advanced levels will definitely learn about these several first 6 sessions that I've mentioned. And also, as I mentioned, those who specifically join the beginner level program only, they would be working on a predefined research question because it's just for 2 months. Now, why did we put this option of a beginner level? curriculum or a beginner level for the research fellowship program. We have received a lot of queries from different universities where students are supposed to work on a mini project, or they want to have a research project, or they want to get an experience with a specific type of omics analysis during their internship, and they have to submit their certificate for internship or mini project, or submit a mini project to their college for their academia. So there could be n number of reasons, and there are also high schoolers who want to work on a research project, present it in an exhibition or a science fair, and that helps them get away or submit their application or complete their application to get to their dream college. So there could be a number of reasons, because time is a limit here. You just have 2 months that you can work on. That's why we have put together some predefined research questions for you. So we have these research questions that we are already working on, and also we have the pre-analysis of these different components already done, and you would be responsible to carry out a literature review and work on the analysis and interpretation, a customized analysis that will tell you that you have to work on. So you get the halfway done and half work done, and then you have to work on the analysis and the interpretation. Now I'll tell you about some of the predefined research questions that we are proposing today for different research specialization tracks. You can pick any one of these predefined research questions if you're interested to get a taste of the different research tracks or different types of omics analysis for your experience or project work. The first one is oncology. It is one of the exciting areas of research to understand how the specific genetic mutations can lead to resistance to cancer drugs. And here we are looking for ways to develop a targeted therapy to overcome the resistance. Another focus is to identify the unique molecular signatures and the biomarkers that are associated with the progression of breast cancer, which can lead to more personalized treatment approaches for patients. Now, these are the two topics for oncology. I've just specified 2 such questions for each of the tracks. But if you wish to work on a different specific track, we can have a call and we can let you know about the other topics that we are working on as well. The next is precision medicine. So precision medicine is all about tailoring treatments to individual patients. And we are researching how to identify the genetic and the molecular markers that can predict how a person would respond to specific medication. And this can lead to highly personalized treatment plans. Additionally, we are investigating the genetic factors that affect drug metabolism, helping us optimize the drug prescriptions. Next is infectious diseases. So here we are exploring the genetic diversity and the evolution of the influenza virus in different regions. And this knowledge can help us design either better vaccines or antiviral drugs, a better therapeutic approach in short. And we would also like to study the transmission dynamics of diseases like malaria, aiming to reduce their spread in endemic regions. So that was about these 3 different tracks and what topics are we focusing on. The next one is neuroscience. So in neuroscience, we are focused on understanding the underlying molecular and cellular mechanisms of neurodegenerative diseases like Alzheimer's or Parkinson's. And our goal is to develop innovative therapies to slow or halt disease progressions. And we are also exploring how genetics and epigenetics influence brain development and the risk of mental health disorders like schizophrenia, aiming to identify early biomarkers for early intervention. Another field is agriculture, where we want to look at ways to optimize crop breeding using genomic data. And also, we are studying the genetic factors and metabolic pathways that make the plant resistant to pests or diseases with the aim of promoting sustainable and eco-friendly farming practices. Next is metabolic disorders. Metabolic disorders research centers on understanding the molecular factors that contributes to condition like insulin resistance in type 2 diabetes. So we are working on targeted therapies to improve insulin sensitivity. And additionally, we also want to investigate the metabolic underpinnings of obesity and its related health complications, such as fatty liver disease. And our goal is to develop personalized interventions for weight management. Last but not the least is space omics. Why space omics? So in the fascinating realm of space omics, we are researching how extended space missions affect the human microbiome. And this knowledge can help us develop strategies to mitigate health risks associated with microbiome changes during space travel. We are also exploring the genomic and the proteomic changes in organisms exposed to extreme conditions in space. And this information can, this specific information would inform the development of biotechnological solutions for space exploration as well as colonization. These were some fields that I wanted to discuss, some topics that we are working on and have plans to work on. And also, there are other fields that I have not discussed in greater detail. But if you're interested, let us know. You can email us. We can have an exploratory call with you and discuss. And those fields would be around cheminformatics that we can discuss specifically around cheminformatics. So those were some of the topics under the beginner level that I wanted to discuss. Next comes the intermediate level curriculum. Again, to mention, the ones who take up intermediate or advanced level would definitely have the access to the beginner level sessions that are discussed, but the one who takes up the intermediate level has to propose his or her own research proposal, has to propose his or her own research question. So if you already have a question in your head, but you want to discuss with us, frame it a little bit, and then you have a proposal ready, you submit it to us, and then we start working on it. So you perform the literature review, you look for the data set, you explore the data set, and you process all, and you go ahead with all the pre-processing steps and the analysis. You'll have all our support. We'll be assisting you at every step, but it's just that the research question is yours. The intermediate level is for 3 months. You get more time to work on your research project. And because you will get more time to work on your research project, that's why you have some more sessions to refer to get that learning, to learn about specific types of omics analysis. Now, these training sessions would basically help you build your concepts. So these training sessions are actually a series of lectures and also some coursework that you have to complete that will assist you to complete your research project. As I mentioned, the outcome of the beginner level could be a research project, a mini project. But the outcome of an intermediate level could be a dissertation project. So a lot of bachelor's and master's students have this criteria to have a dissertation project submitted to their college or university to get their final degree. So the intermediate level would cater to that need, where you can work on your dissertation project. Obviously, because you get more time, so you can work on this specific, or you can work in this specific direction. Now let's discuss what the training of the intermediate level would look like. So as I mentioned, that you have to pick your own research question. That's why it's very important for you to understand the different research specialization tracks. Today, I just introduced you to the tracks. I did not discuss much in depth about each one of these. So in the 7th session, we will discuss about each research specialization track in detail. And based on your interest, you can pick the one that you want to work on. We will explore the unique challenges and the opportunities within each specialization that will allow you to align your research to focus with your passion. And the 8th session would be around omics databases and data retrieval. So let's say you have a research specialization track that you want to work on. For example, you want to work on oncology. Next thing is to, first of all, pick up a specific type of interest of omics data that you want to work with. Let's say amongst all those different omics data types, genomics, transcriptomics, metagenomics, and single cell or epigenomics, I say I want to work with transcriptomic data. So the track is oncology. I want to work with transcriptomic data related to oncology. It could be any type of cancer. Let's say I pick liver cancer. The next thing would be to extract the data, to look for data. I want to look for liver cancer data sets. So it's very important for you to learn what are the different omics databases from where you can retrieve the data. So in this session, we will introduce you to various omics databases and the importance of data retrieval. Now, data retrieval is not very easy. Different databases have data uploaded in different formats and in different ways. So how do you retrieve the data? How do you clean the data? Those are some of the things that we will be discussing. So here you will learn how to navigate and extract the relevant data from the databases to support your research. Now, there are different omics tracks that you could pick: genomics, metagenomics, or transcriptomics. Now, here you have a choice. It's not that you take all the sessions. I said that I'm interested in transcriptomics. So if my interest is transcriptomics, I'll just take 11th and 12th session. If your interest is metagenomic, you just take metagenomic session. Why we have put these sessions here? So that we can guide you in more depth how to work with a specific type of omics analysis. If it is genomic data analysis, that is your interest. So we'll teach you, or we'll delve into the genomic data analysis that will focus on variant calling to identify genetic variations. And you'll also learn how to perform functional annotation to understand the implications of genetic changes, and the phylogenetic analysis to trace evolutionary relationships. So we'll discuss these different possible analysis that comes under genomic data analysis, or that is possible with genomic data. Okay, next is metagenomic data analysis. Now, metagenomic data analysis is essential for understanding the complex microbial communities, as I mentioned. And because you now want to analyze or understand the composition or the abundance of these different microbial communities, it's important for you to learn about different tools and techniques that you could use to analyze microbial data. Now, these tools could be like Data2 or Kind2. Those are the two specific pipelines that we would be focusing on that will help you understand how to process and analyze data, allowing you to explore microbial diversity, taxonomic classification, alpha, beta diversity, etc., etc. The list goes on. And in the next sessions, session 11 and 12, you will learn about transcriptomic data analysis. So RNA-seq is a powerful tool for gene expression analysis, and many of you might have a good hands-on with this type of analysis as well. But if not, then these sessions will help you learn how to explore the data. So once you have the data with you, you need to perform exploratory analysis to understand the data set in your hand. We'll discuss various statistical methods for differential gene expression analysis in this session. The next session is on differential gene expression analysis, where we'll dive deeper into the analysis of differential gene expression. That's a very critical component of omics research. You'll learn how to identify the genes that are significantly upregulated or downregulated in different conditions, which is crucial for understanding biological processes. So the intermediate level sessions will build on the foundation that you establish in the beginner level. That allows you to specialize in a specific research track and also gain in-depth knowledge in omics data analysis that is valuable for your research fellowship. So that was all about the intermediate level. The next level is the advanced level. So for those who are aiming to publish their work, the advanced level is where it all comes together. So in the intermediate level, you could just cater the need of submitting a dissertation to your college or university, or maybe work on a research project if you're a PhD scholar. But if you have a requirement to publish a paper, or it's very obvious that it's not possible in a span of 3 months to learn, work on a project, and then also publish your work. That's why the advanced level was developed or put together. That will help you. Advanced level is for 6 months, first of all, and this specific level will help you with the specific set of tools and techniques that you require to perform a good research work, and also have the opportunity and the time definitely to prepare a manuscript that could be further published. Now, why I mentioned the advanced level will give you the access to these sessions that would help you in advanced research, when I say. So we learned about different omics analysis in the intermediate level. It's very important for you to also learn about the tools and the techniques that could be used to perform analysis with that specific type of data, or perform advanced analysis, I'll say. In the intermediate level, you learned about genomics, metagenomics, transcriptomics. You need to pick one track of your interest. Advanced level will help you learn about single cell data analysis or single cell transcriptomic data analysis. The first 3 sessions of the advanced level are around omics analysis, and then other tools and the techniques. The 13th session is about pathway and functional enrichment analysis, where we delve into advanced RNA-seq analysis, where you want to learn about the pathways or the functional enrichment of the differentially expressed genes. You want to understand which pathways your differentially expressed genes affect. And also you want to understand the enriched, along with the enriched pathways, the functions of the function of your DEGs, providing valuable insights into the underlying biology. The next 2 sessions are on single cell data analysis. It is one of those techniques or analysis that a lot of researchers are working on, but because the sequencing of this type of data is quite costly, that's why it's not a routine, or you'll not find a lot of single cell data available. But there are a lot of databases from where you can extract single cell data, and we also have an amazing pipeline on single cell data analysis. So for this specific track, we have put 2 sessions. So here we will explore the databases. First of all, how to retrieve the data, what are the different types of techniques to get the access or retrieve the data and work with the specialized data. And we'll understand where and how to obtain the single cell data that is crucial for advanced research. And building on the previous session, we'll then dive into the analysis of single cell data, focusing on cell type identification, differential expression, trajectory analysis, and advanced sort of analysis, and also learn about or understand about cellular heterogeneity and dynamics. So that was about the omics analysis, different types of omics analysis. Now, if you're not interested in single cell, but if you want to do genomic data analysis, you just refer to the genomic session and not the single cell one. That's an option given to you, but we definitely will recommend that you take up these sessions so that you learn about the different types of omics analysis, and maybe in future you can work with any other type of analysis. And then from the 16th session onwards, we are talking about the most talked about topics, data science. So data science will here introduce you to the data science principles and the techniques that are specific to omics data. You'll explore data pre-processing, feature engineering, model selection, and for more advanced data analysis, you can refer to it. We'll also teach you about machine learning, different types of machine learning, unsupervised analysis, supervised analysis. What are these different tools that could be used for both of these different types of analysis, and how do you work with them? We'll teach you through the automated pipelines and also through script. So that was about unsupervised and supervised analysis of machine learning. And we have 3 sessions on supervised analysis, taking each algorithm at a time, and then following the process where you have the data and then you prepare your data and then you separate it into training and test set, and then you apply different algorithms, check the accuracy, evaluate your model, and then you deploy it. I've just given you a very brief overview. So we'll discuss the entire flow, and also we'll discuss about each and every tool. Why is it used? How is it useful? When to use it, etc., etc. Okay. Now in the next sessions, we will discuss about R and Python. Not everyone is a techie here. Not everyone wants to get into the coding, but those who are interested to learn R and Python for biomedical data analysis, I think the advanced level is best for them, because learning R and Python will, I'll say, make you a full package of where you know about the different types of analysis, and also you know about different libraries that could be used for this analysis. So it will help you build a good research project, but also gain a good experience alongside. So in the next session, we introduce you to the languages. First of all, it's not important that you have to learn R and Python both alongside. You can pick one language at a time, either R or Python. We'll give you the resources to learn any of the languages, R or Python, for biomedical analysis. If you want to learn both of them, perfect. So here we'll discuss about R and Python. We introduce you to the languages, how to process data, how to perform exploratory analysis, how to perform statistical analysis. Also in the session name that we have not mentioned, but in the coursework, you'll learn about machine learning as well, how to apply or build different machine learning models using R and Python. Now we get one question a lot of times: how many hours you need to dedicate for each level? See, the number of hours, it really depends on you. Now, there are some people, if you have a non-bioinformatics background, you might have to give more number of hours. If you are a working professional and you have time on weekends, you can give more time on weekends, and you can accommodate your timings on the weekdays. And if you are a student and you have more time on the weekdays as well, then you can accommodate accordingly. There are some students who are fast learners, so you might require less time. So accordingly, we provide you with the resources. It completely depends on you how much time you take to go through these resources. But yes, we'll do provide you with deadlines to make sure that you achieve your mark on time. And we'll keep assisting you with your questions, clarify your doubts, and take you a step ahead towards your research. So that was about the different levels: beginner, intermediate, and advanced. If you have any questions, you can put in the chat box, and I'll now take you to the OmicsLogic Learn portal, where I'll just show you all these different levels and what resources, what access to the resources you have. Any questions? You can also unmute yourself. All right, so if there are no questions, I'll now proceed ahead. So this is the program page that you might have seen on the social media as well. So once you're on the OmicsLogic Learn portal, you will find this page under the Programs tab. This program page is very important for the program because you'll find all the associated resources that I just went through: the sessions, the recordings, the lectures, the coursework, everything on the program page. How can we get the data if you're interested in a particular topic? That's what we'll teach you, how to get or extract the data from databases or repositories for a specific topic that you're interested. Dohan is asking how we will do our analysis, as we know that bioinformatics need large computer resource, which maybe our machine can't run. Dohan, the answer is T-Bioinfo platform. I'll just take you to the T-Bioinfo platform where you have these pipelines. You just need to extract the data through links. You can refer to the specific data set that you want to work with, upload your data here, or put the links to that specific data set from the data repository, and everything would run on server. You will be given 5 TB space where everything would be done from pipeline processing. You get the results, you just have to download the results and interpret them. Okay. I'll take you through the platform. Just give me 5 minutes, 5 more minutes. I'll just take you to the platform. It's not an online machine. It's a server, basically. It's an online server, and you are allocated space on the server, and you can run the educational pipelines to learn, first of all, how the analysis is performed, and then you perform it with your own data. So that is how it would work. Okay. Now, I'll tell you how to get to the program page. I'll just show you the different components of the program page, and then I'll take you to the resources that you get an access to with the program. When I say resources, I am referring to the coursework and this platform, T-Bioinfo platform, to process the pipelines. So there are two things. Don't get confused. OmicsLogic Learn Portal, that is like your training guide. You have all the resources here to learn from. Then you have the T-Bioinfo platform to perform the practical hands-on. So you go to the programs, and here on the second page, you can also put in the search bar, research, and you'll get this program page. Now on this program page, if you scroll down, you have the specifications of the program. Then you have these 3 different levels: beginner, intermediate, or advanced. You have to pick up one of the levels based on your research interest. And I'll just take you shortly how do you register. On the program page itself, you will find these different levels. You pick the specific level that you want to enroll with. And then the moment you do so, you'll unlock that specific level for yourself. So here you have these different sessions. These are the sessions that I took you through when I was going through the presentation. And here you will find the link to the lecture. You'll find the link to the resources, which will include coursework, the courses that you have to go through, supplementary material, additional material, link to literature or research articles or additional material that you can go through. All right. And you just have to watch it, complete the material, and work on a research project. We will be meeting every week where we will discuss your progress. We'll take up your queries. We'll take up your queries and rectify them and help you proceed ahead with the next step. Now, what are the resources that come along? I mentioned coursework. So you would pick one type of omics analysis. Let's say you want to work with transcriptomic data. You pick one type of omics analysis, learn how that specific type of omics analysis is performed. And then when you have to select a research question or an area that you want to work on, then you pick up a research specialization track. If you want to analyze RNA-seq data for an Alzheimer's condition, or for a specific type of cancer, or for metabolic disorders, diabetes or obesity, that way. So now let's say you want to learn about transcriptomics. Before I hit on the transcriptomics coursework, let's jump to the initial step. Let's say you are a newbie and you don't have an idea what specific type of omics do you want to refer to. For you as a newbie, we'll take you through the first course, that is Introduction to Bioinformatics. Now, this specific course will introduce you to the different fields of omics: genomics, transcriptomics, metagenomics. It will help you understand what this field is all about, what this field includes, what type of data has to be analyzed in this field, and different applications. So this is a coursework. These are different lessons. You open up a lesson. Inside the lesson, you have context, you have images to supplement your understanding, you have videos to watch, you have quizzes and assignments to answer to check your knowledge. So that is how a lesson is built. Okay. Now you go through this coursework and then you let us know which specific field you're interested to work with. Now, the next step is, let's say you specify that transcriptomics is that area that you want to work with. Sorry for the typo. Okay. You mentioned that you want to work with transcriptomics. So the next step would be an advanced training. You go through the transcriptomics coursework. Now, in this coursework, we start from the very basics. Even if you don't have a background with transcriptomics, it's perfectly fine. We'll start with the very basics. We'll start with the concepts of what is transcriptomics, how transcriptomic data is sequenced, what are the different sequencing techniques, what are the different sequencing types. And then once you have this data, how do you clean the data? What are the steps involved to clean the data, pre-process the data, and then perform exploratory or downstream analysis? What is the flow? And also, what are the different tools that you have to use? Okay. So inside these lessons, you have the concepts explained, and you also have the practical hands-on explained. So you have the steps that you need to perform. So if I am not wrong, Dohan, yes. Dohan asked how to perform the practical hands-on. Okay. We definitely know that your system won't support it. Not everyone has a high computing system. Actually, no one has it with them at their places. Sorry. So in the coursework, you have these lessons where you have the steps specified. You have a detailed description on what tools are to be used, what those tools do, and what are the different steps, what are the different parameters, and then how to complete the processing of the pipeline, and then what results you get and how to interpret them. So that is one thing. Now, everything from the very beginning, understanding how the data is pre-processed to exploratory analysis or downstream analysis. The next thing is you learn how to perform statistical analysis or tests, differential expression analysis, regression analysis, machine learning, everything around transcriptomic data, right? Once you have completed the coursework and once you have a good hands-on the specific type of data analysis, the next step is just to get an overview of how the concepts that you learned could be applied to answer a research question before you really start working with your research question. So you went through the coursework. First of all, you identified your area. After that, you picked up a specific coursework, learned the analysis. And this coursework would also be supplemented with mentor lectures. You watch the lectures for each step, understand the flow, and then you also go through some case studies. These case studies are not very lengthy, they're very short case studies, but it will help you build a concept. So after you know the concept, how to use it to apply to a research question, that is what you will learn. Okay, so that was about the coursework that you have to complete as a part of the program. That's actually not a lot of coursework. It's definitely a gradual learning process. You learn a thing and then you apply it. So that's the way it should work. I mentioned about the T-Bioinfo server, so let's have a look at it. This is the practical hands-on platform that you would have to use. Now, definitely there are three things that we would refer to. We'll refer to open databases to extract data. We'll also refer to the open or publicly available tools and techniques that you can use, or publicly available softwares that you can use for analysis. And we'll also explore the T-Bioinfo platform. So we'll explore all the different aspects of bioinformatics, tools, softwares, and techniques in R and Python as well that you can use to analyze your data. So when it comes to the T-Bioinfo platform, there are different areas of analysis on the platform. Transcriptomics, genomics, single cell metagenomics, virology. Then you have the phylogenetic analysis here. For machine learning, you have pipelines. There are several pipelines. I'll just take a few minutes of yours. I know we are at the time of ending the webinar, but I'll just take a few minutes to give you a quick hands-on with what these pipelines are and show you how to process them and how to interpret the data. So here you have these drop-downs. These drop-downs have some case studies or pre-built pipelines. We call them demo pipelines. Why they're called demo pipelines? Here the data is already uploaded at the backend, and you just need to process the pipeline, look at the result and understand what these results are and learn to interpret the result. It's not difficult on our platform to take the data and process the pipeline. but it's important for you to understand how to interpret the result. So how do you upload your data? This is a usual pipeline. You simply upload your file. It could be a TXT file. It has to be a TXT file, or an SVL link, or an HTTP link, or an FTP link. So Dohan, you were asking, how do we do that? We can also refer to these different links that we can get from the databases. Okay, and then we upload our data and then we process the pipeline. I'll quickly take you through one of these pipeline processing. So this is a pipeline. I can simply start the pipeline, use some specific algorithms, what these algorithms are, why am I using them, what parameters do I need to put, everything we'll teach you in the program. Right now, I just wanted to show you how easy it is to process a pipeline, and then you have your results that you can interpret. You get the results here. You can click on them. It will download. These are zipped files. You unzip them and then you explore them. And you get a dashboard where you can change the parameters and see how your results change with change in parameters. And then these are the interactive plots that you get through the pipeline that you can interact with and interpret your results. So that was a very quick and a brief demonstration of the T-Bioinfo platform. There are various pipelines that we could use for different types of analysis. We'll be talking more about them once you join the program. And with that, I'll pass on the stage to Shri Gauri to quickly show us the outcomes of the program. And then we'll discuss the registration process, and then we'll be good to end the session. If there are any questions, please feel free to put in the chat.
Thank you so much, Sonalika. I'll just quickly share my screen. So the participants have any queries, please feel free to put it in the chat, and along the way, we'll be able to answer your questions. So Sonalika gave a very comprehensive introduction to the various specialization tracks that are there as a part of the research fellowship program. And now we'll take a look at some of the ways that the students have leveraged these training program and the resources to come up with various outputs. So in this case, as you can see, the research program was integrated as a part of the university curriculum, which was part of the Systems Medicine graduate program at Georgetown University. And the training program ended with students completing a diverse range of research projects on machine learning using Python to address various research questions in biomedical research, such as infectious diseases, metabolic disorders, neuroscience, oncology, and so on. Further, students have also went on to participate in poster presentation competitions that we conduct annually, which is the Omics Research Symposium, wherein international experts will attend, review your research project, and provide feedback on how you can improve it further. And there are also students who have went on to publish their research project in open access journals, such as Prithvi Waja, who is a biomedical science student who is interested in immunology research specialization. And let's say if you're someone who is interested in cancer diagnosis and treatment, then you can also opt for the oncology specialization track, like Sinead or Randall did, who went on to publish her research project. That's part of a university at Columbia. And there are also students coming from the high school background. For example, Modano Souza, who presented his neuroscience project at the Regeneron International Science and Engineering Fair, for which he won the Potamkin Prize. And presently, he is enrolled in an undergraduate program at Harvard University. And we also have students who complete their research project to participate in international conferences, like Priyanka Sharma and Kriti Pandey. They were from biotechnology background. And these projects can also be showcased as a part of your university application. Like some of our students did, such as Jalara, Mehra, Anisha, they all went on to showcase their research project in their resume as part of their university applications. Now, taking the example of Dilara itself, if you're someone who's interested in astrobiology and space omics, we had a lot of students from that background who are still actively passionate about the field, and you can find a lot of their session discussions as part of the student club that we have on the YouTube channel. Now, moving on, if you're someone who is interested in infectious diseases, that is also possible. And here is an interesting example where the faculties and researchers collaborated through our research program to publish their research work. And this particular example is part of the Nebuti Memorial Institute for Medical Research, which is part of University of Ghana. And let's say you're someone with a passion for science and business, then it is also possible to pursue an entrepreneurship and business innovation track, like a team of industrial researchers from Biologic Support Services, which is a biotechnology company in Nigeria. So they specialized in the microbiome metagenomics, and they developed a microbiome test kit to improve the fertility of women in Nigeria. And the interesting aspect is that such entrepreneurship tracks are not just pursued by researchers, but by students as well. Here is an example of a group of undergraduate and postgraduate students from India who developed an early detection kit for Alzheimer's disease. And now, to regularly stay updated about our ongoing research project opportunities, student publications, exciting job opportunities, latest research articles, and there in bioinformatics and data science, we encourage you to follow our LinkedIn group on machine learning and data science, where you will get frequent updates as well as student updates. So that is all that I wanted to discuss about the outcomes that are there as part of the research program. And I'll share the link to these groups in the chat box so that the participants can join the group. Yes. So now I'd like to pass on the stage back to Snalika, who will walk you through the registration process and how one can. So over to you.
Thank you, Shrikari. Okay, so now how do you register for the program? So to register for the program, you need to go to the program page, go to the programs tab. And there was a question, is it easy to access the OmicsLogic portal? Yes, it is very easy. But if you have any sorts of technical problems navigating the program, you can anytime reach out to us and we can have a call with you one-on-one where we can tell you how to navigate the program, the portal. All right, so once you're on the program page, you scroll down and you have these three sections: beginner, intermediate, and the advanced level. And based on whichever level that you are interested to enroll in, you can pick that level. Based on the country that you are present, let's say I'm from India. So if I select INR, I'll get these three different levels. We are offering scholarships at the moment, and we had a lot of queries in the chat box as well for the scholarships. Yes, we are offering scholarships. You can reach out to us on communications@pine.bio and just put your educational background, or if you are working somewhere, you put your research position, name of the institution or the university, and then you put the scholarship reason, right? So there's a form that you have to fill for the scholarship. So you email Shrigori, she can put her email address in the chat, and you just fill out that form. We'll review your application, and then we'll review, and then we'll give you a discount code. You take that discount code, click on the buy now button for whichever level that you want to enroll in, and then you put that specific coupon code over here, and then you click on the apply button. And once you do that, the amount would be deduced. So that is how you get the scholarship and use that coupon code to put here, right? So if you're seeking for a scholarship, please feel free to reach out to us. Shrigori, please put your email address in the chat box so that we can help you get a scholarship and start with the program. These scholarships are available for students from low-income countries and also from students from specific institutions in universities. That's why I asked you to mention your institution or university as well. Now, just to discuss about the specificities for each of these different levels that I discussed. I'll repeat once again. For the beginner level, you get an access for 2 months. Intermediate gives you an access for 3 months. Advanced gives you an access for 6 months. In the beginner level, you will be working on a predefined research question. We'll give you some research questions. Either you can pick from it, or the research projects that are ongoing in the company, you can become a part of that project and have a customized research question that you can work on specifically for your research project report. Okay. The outcome of the beginner level would be a research project report that you can submit to your college or have an experience with. And also high schoolers can enroll in the beginner level. They can take the other levels as well. But if you wish to work on a mini project for an exhibition or a fair, then you can pick the beginner level. And then comes the intermediate level. That is for 3 months. Here you have to propose your own research question. We'll assist you work on the research question if you don't have a research question. And then you can also work on the specific research question for 3 months. And the outcome of the intermediate level would be a dissertation maybe that you can submit. It could also be a poster. We can help you with creating the poster of your specific research book that you can present maybe in conferences. Or we can also assist you for these posters in poster competitions in your campus, university, or college. And then the advanced level is specifically for those who want to get an advanced hands-on with the tools and techniques of data science that would be used to apply to their research question. And they want to have a research publication out of their research work. So they can pick the advanced level. That is for 6 months. So based on your requirements, you can pick the level that you want. Again, for scholarships, you should email Srigori on communications@pine.bio, and we'll review your application and give you a workable report. All right. Are there any questions?
Just a clarification here that. So most of the participants, I believe, have already submitted their applications through the form. So we'll review that existing submissions that you have. And for those who are new and joining us for the first time and have not submitted, please register through the form. I'll share it once again in the chat box. And so after reviewing the submissions, the selected candidates will be emailed with the scholarship code that will be provided. Just that clarification. And if anyone has any further queries, let us know.
And feel free to connect with us on LinkedIn. We have the groups by OmicsLogic. We keep on updating there about the scholarships available, about the ongoing research work. So if you go on LinkedIn, you have the pages here, OmicsLogic. One is this page, and then you also have a group that you can join. Just write OmicsLogic and feel free to connect with us on LinkedIn. Srigori, you were mentioning something. Please, over to you.
Yes, we have one query from Miracle that he is a graduate of biochemistry and he is not currently enrolled in any institution, but he needs the research experience for his application process. So can I qualify for the scholarship regardless?
Yes, definitely. You will qualify for the scholarship. Please mention or please share your resume with us. You can. We just want to look your educational background, look at your educational background, and also look at the previous research projects that you have worked on. That would help us.
I've shared the email ID in the chat box. So if you have any queries, you can drop an email there and our team will get back.
Perfect. Thank you so much, Srigori. Do we have any other questions? What percent of discount? There are different scholarships that we are offering based on the background, the student background. So once you send your application to us, we'll let you know what scholarship do you get. You can share it directly with Shri Gauri on communication@omicslogic.com. Yes. All right. Thank you everyone for joining today's session, and we'll see you in the program. Once you join the program, we'll have an orientation session to get you started, and we'll keep on meeting every week to look at your progress and help you with the next steps. See you all in the program. Bye-bye.